the T cell populations as belonging to three primary groups. We call them cytotoxic T cells, or CD8 cells. We call them helper T cells, or CD4 cells. And then we refer to the T cells as memory cells. So those are the three groups that we introduced last time. We said that the primary functional description of the T cell populations is cell-mediated immunity. And this picture illustrates what we mean by cell-mediated immunity as well as any I've ever seen. Because you can see the T cells being, you can see that these T cells are mediating a cell-to-cell interaction here. And so, as we finished up class last time, we were talking about the differences in these two. But I did want to emphasize to you one more time, because it's going to come up on a couple of occasions today. In fact, on the next picture, I want to emphasize to you once again that I don't really like the description being limited to cell-mediated immunity here, because if you talk about cell-to-cell interaction here, it gives the impression to me, to students, and to me all of my life, I've wondered why these things held on like they did. It gives the impression that this is all the T cells do, that they come in contact. They're a cell-to-cell interaction, and that's true, they do. But you know, B cells do that too. They have antibodies with variable regions on the end of them that come in contact with antigens on other cells. That's what the T cell's doing here, come in contact with the antigens. So I have to teach you these things. I don't like them, but I have to tell you what your textbook says about these T cells. So there it is, they're cell-mediated. What we're going to see on the next picture is the T cells, in addition to facilitating what we described last time as cell-mediated immunity, they also release chemicals, and we call those cytokines. And the cytokines exist in a number of varieties. They do a number of things, including activating other T cells, activating other B cells, causing proliferation of these cells, causing antibody release, and a whole host of other things, causing B cell numbers to increase, etc. So cytokines are very important molecules released by T cells. And normally it's the helper T cells that are doing this. So that's what I'm going to show you on the next picture. All right, so here comes the intro. Last time we said this: cytotoxic T cells are, along with their cousins, their first cousins, called the NK, or natural killer cells. These guys are really the prototype of what we mean when we say cell-mediated immunity. They recognize class 1 MHC molecules. These are molecules produced by MHC genes within most of the cells of your body. They produce antigens that cause the immune system to recognize you as you. That's what these MHC molecules are. The cytotoxic T's recognize foreign antigens associated with these class 1 molecules, and when they do, they release a molecule called perforin that cuts holes within the membranes of infected cells and then causes them to be destroyed, normally by the influx of water. So by cutting holes in the membranes of the infected cell, it allows the water to rush into the cell, and then it lyses. Okay, so this is the prototype of the CD8 cytotoxic T, and very similar to what their cousins, the natural killer cells, do. Now, the story, to me, gets more interesting here, because the CD4 helper T cells have a little different mechanism of action. In fact, these guys recognize foreign antigens associated with class 2 MHC molecules. It's a different sequence on this molecule. And these guys are made, these class 2 molecules are made typically by antigen-presenting cells. And there are a number of varieties of these antigen-presenting cells. The one that you need to know from me are these, because we've mentioned them so many times. This is a macrophage. It used to be a monocyte. And the macrophages, as you know already, are very good at phagocytosis. They eat things. And so here the macrophage is eating a microbe, and as it does, it cuts the microbe up and then exposes the foreign antigen along with the class 2 molecule to a CD4 cell. Then the CD4 cells are going to release their cytokines and activate an immune response. So I wanted you to see the story. this picture tells the story for you. And here is the macrophage. Here is the microbe being ingested. Now, it doesn't show all the details here, but macrophages have very powerful organelles inside them called lysosomes. Please don't confuse lysosome, a cell organelle, for the lysozyme enzyme produced by the lacrimal glands. Tears have a lysozyme enzyme that's an antibacterial agent for the surface of the cornea. These are lysosomes. They're an organelle in the cell that contain digestive enzymes, very powerful digestive enzymes. In fact, most of the lysosomes in macrophages have about 20 different varieties of these, and it is very difficult to explain in chemistry and molecular biology, however you want to put the categories, why the digestive enzymes of the lysosome don't digest their own membranes. They're very powerful. So what happens is the macrophage ingests some particle, and so it now appears inside the cytoplasm of the cell in this vacuole, encapsulated. And the encapsulated vacuole then fuses with the lysosome, and the lysosome pours the digestive enzyme into the vacuole containing the microbe. And then it goes to tearing it to pieces, enzymatically tearing it to pieces. And as it does, it releases a number of antigenic sequences that are now non-infectious, if you will. They're not going to do anything to us. It's being destroyed here. The beauty of the destruction, however, with the antigen-presenting cell, I said it that way on purpose, a macrophage that's an antigen-presenting cell. The beauty of that destruction is the antigen then is associated with the class II MHC molecule on the surface of the macrophage. This is a beacon to identify the antigen for the immune system. It is presenting the antigen to the immune system. And there will be no doubt in the system a CD4 helper T cell that will recognize, because it knows about the class II molecule, and now it sees that foreign antigen and it says, that does not belong. Now, it is interesting to note here, and I want to point it out again to you. I'm going to say this again with the antibodies as well. It is interesting to note that the presence of this antigen associated with the class II molecule did not create or stimulate in any way the creation of this CD4 cell. This CD4 cell that recognizes that antigen was already in the system. Now that's a problem in logic. The system, the immune system, is set up this way, both for the antibody productions and the antigen recognition molecules that are present on the T cells. It's set up like this. The ability of the cell to recognize the foreign antigen is present before the antigen is. The system is making these cells with these abilities before they ever see the antigens. So you have scientific notation numbers, abilities to recognize antigens in your system, most of which you'll never see. But you have the ability there to recognize it should you get exposed to it. Fascinating to me. The helper T cell recognizes the antigen. In response to that, the helper T cell releases molecules that are called cytokines. Things, when I say that word, you should think to yourself, interleukin-1, interleukin-2, colony-stimulating factors. These cytokines can do a number of things. They can T cell activate, they can T cell proliferate, they can cause increases in other white blood cell counts. Okay, in this story, which is one of many, the cytokines release will then cause the proliferation of B cells. Colony-stimulating factors, that's what these are. B cell, colony-stimulating, lymphocyte, or agranulocyte, colony-stimulating factors, that's what these guys are. They stimulate the B cell to proliferate. So, exactly what B cell are you talking about? So, here's the next step in, you've got to be kidding me. This B cell here is called a B cell, and you should think in these categories already. This guy is called a B cell because it's not a B lymphocyte anymore. It's a B cell now because it's producing antibody. In just a moment, after this B cell guy is activated, we're going to take this one step further. The step's not on here, it's on the next picture. Let me just go ahead and show it to you. In just a moment, we're going to do this. One more. Yeah. In just a moment, we're going to do this. 2 slides away. The activated B cell now is going to now change in name. We're going to call it a plasma cell because it's going to be releasing these antibodies into circulation. So this is the stepwise function of the B cells. All right, now here's the "you've got to be kidding me" part. The cytokine population here is going to cause increases in the B cell numbers. It's going to cause them to proliferate. But not just any B cell population. In fact, in the same way that the T cells recognize this antigen, there are already B cells in the system that have been producing antibodies with variable regions on them that recognize the same antigen. So here's what I'm telling you now. Not only were the T cells already there, but the B cells were already there. And not only were they already there, but they were already producing antibodies that could recognize that specific antigen. So this cytokine population activates a B cell population that has the ability to recognize the same antigenic sequence. Collectively, what I have just described for you, and I've got it written on here, what I've just described for you is a monoclonal immune response, one clone of cells responding to a single antigen. So the B cell population now increases, and these antibodies will ultimately be released and go about binding to these microbes, the antigens that are on them, and causing clustering, agglutination reactions, activating complement, a whole host of other things. I'm going to give you some of them coming in order to destroy that pathogen. It's phenomenal, absolutely phenomenal. So this story is intended to link them all together. The macrophage, the MHC molecules, the recognition of the helper T cells, the release of cytokine, the activation of B cells, it's all here. This slide has really most of the really cool parts of the immune system story, B and T cell interactions. Now, as I said when I started class, we say T cells are for cell-mediated immunity. We say B cells make antibodies. They're for humoral immunity. They release molecules. But the truth is, this B cell population that recognized that antigen could have done his thing without the activation of the cytokine. In other words, I would have called this a cell-to-cell interaction. Okay, but it's okay. It's okay. I'm not mad about it. It's just a little frustrating because it limits students' understanding, I think. These B cells can, in fact, act alone. They can recognize foreign antigen sequences. Normally, the CD4 populations are responsible for releasing cytokines and activating these B cells. And the response that I just described for you is a monoclonal response. But we should extend that. Because when this happens, right here, it's shown on this picture too, when this happens right here, when this microbe is actually cut up by the lysosomal enzymes in the cytoplasm, when that happens, notice that there will be more than one antigen. This one shows 2, but there are likely to be dozens, maybe hundreds of different antigenic sequences that have the potential to be associated with the class 2 MHC molecules. Now what's going to happen in your imagination, if instead of this triangle being here, I put this little semicircle here, well, here's what will happen. There will be a different helper T cell population whose class 2 molecule will have a semicircle shaped antigen here. So a different group of CD4 cells that have a recognition capacity for a different antigen. And as they recognize that different antigen, they will produce cytokines that will then increase the numbers of B cells that have regions on them that recognize not the triangle one, but the semicircle one. Are you guys following me? So normally, an immune response is not like this. This is a monoclonal response. The system is responding to one antigen. But when the lysosome busts up the whatever it is, normally, when this happens, there will be a number of different antigens exposed here. And the system will recognize all of them. So an immune response in our bodies is almost never a monoclonal response. It is normally a polyclonal response, especially if you're talking about dealing with a microbe. Now, this phenomenon of monoclonal and polyclonal responses is now something that we take advantage of in immunology, in immunocytochemistry studies. I used it years ago in doing research things. It is arguably the most interesting area of human medicine today, in that what is being done now, because we have all this recombinant DNA technology available to us, we can insert genes into bacteria, etc., and make them make stuff for us. What's happening now in the medical community is the development of specific drugs that are monoclonal drugs. Things that can attack, things that can go about helping in our system, getting rid of things that we don't want in a more specific way. So there are scores of examples of this. But let me just give you one way this might work. Let's say you have a cancer cell in the human body. Normally the cytotoxic T's recognize it, release perforin on it, and destroy it. But what if you could take that cancer cell and isolate antigenic sequences that are not cancerous on the cell and amplify them? And you could then take those antigenic sequences that are non-cancerous and give a vaccine, if you will. So y'all should hear Gardasil. I'm describing it right now. You give this vaccination of all these non-harmful antigenic sequences into the system to cause a bloom, a huge increase in the numbers of T and B cells that recognize that particular antigen. Now what's going to happen? You have taught the immune system now how to target and go after a specific antigen on those cancer cells. Turn that sucker loose. All over the body now, your immune system is going after that antigen. So, what an incredibly powerful way to do this. And it's showing up now in a lot of different places. My father-in-law took a test drug. It did not work for him, but they're getting better and better. He took a test drug for pancreatic cancer. There are vaccines now available. In Texas, they passed a law a few years ago that all young ladies had to have this shot, and then Rick Perry vetoed it. It was a monoclonal antibody vaccination for female reproductive cancers, cervical cancers in particular, very common in young ladies, with 100% success rates. It's unbelievably effective. So, monoclonal, polyclonal responses, I wanted to pause there for just a moment. In immunology, in the lab, if some of you have the opportunity to take this, I think Dr. Criswell and Dr. Du Bois will be team teaching this, I think, in the fall. There are no immunology lab manuals available on the market, but these two are world-class immunologists. They're going to develop some labs. They've done this before together. It's very cool stuff to work with these monoclonal antibodies. And so you're likely to get some exposure there if you're interested. I used them years ago when I was interested in staining nervous tissue for astrocytes. As it turns out, astrocytes make a protein that's unique to the astrocytes, called vimentin, or glial fibrillary GFAP, glial fibrillary acidic protein. You can buy, through Sigma, you can purchase a monoclonal antibody that recognizes that protein. So we mixed it up in a solution. I cut up a bunch of chicken brains, and I poured the monoclonal antibody all over the tissue. So wherever the astrocytes are, the antibody binds. And then they provide you with a kit that you can put on top of the antibody and make a precipitate out of it. In other words, when you put the next molecule on top of it, it's called an adenine biotin complex kit. When you put that on top of the antibody, it comes out of solution, and you put it under a microscope and you can see it. So it shows you where the antibody bound. It identifies where the astrocytes are. Very cool. It's a process called immunocytochemistry or immunohistochemistry, using monoclonal antibodies to recognize things unique in the brain, in other body tissues, and things like that. So this comes from the knowledge of the immune system. And I like to pause here and really hammer this home, monoclonal and polyclonal responses. All right, now, when you bloom, when you activate a group of B cells, the same sort of things happen with the T cell population. So these B cells have been activated, and when they get activated, here's what happens. You will produce, as a result of the activation of the B cell populations, you will produce a clone of cells. This picture is a picture of a monoclonal bloom of B cells. Now, as a part of that bloom of clones of B cells, you will produce active B cells that will be releasing their antibodies. Not only do you increase cell numbers, but you're going to increase the titers, the amount of antibody in circulation. And remember, we're talking about the specific antibodies that have the binding regions that recognize one antigen, one particular antigen. So lots of these antibodies being released by these plasma cells. But in addition to that, just like the T cells had memory cells, the B cell populations also make memory cells. And for reasons that are still poorly understood, these guys do not become plasma cells. You have large increases in the numbers of these cells. They've produced the antibody that has the binding region for the antigen, but they don't release it. They sit in circulation. They become dormant. Here's the beauty of this. This is what Jonas Salk realized when he first developed, out in San Diego, he developed the first vaccine for polio. And this was the logic behind it. So here's what happens upon a second exposure to that antigen. In a primary exposure, the antibody concentration changes over the course of days within circulation. And this makes perfect sense. Over the course of the first week or so, you're going to get significant elevations in the antibody concentration, and that peaks out at about 2 to 3 weeks. You get a peak level of antibodies. This is normal. You get exposed to something, this is what happens. You get an increase in antibodies. Now, as a result of the populations of dormant or memory cells, on a second exposure, the antibody concentrations increase orders of magnitude faster and higher in a second response than they do in a first response. So notice here, by the time you get to 10 days here, look at the differences in the concentrations of antibodies. It is no comparison. The second response is dramatically higher. This is the picture that I would use in any clinical setting to prove to young parents that they should vaccinate their children. This is the one that I would use. So I tell the nursing kids this. I show them the same picture. And I had a class of students 4 or 5 years ago, Martha Means and Emily Weingart and all that bunch. They took me to heart here, and they produced for their classmates this picture laminated cards for all of them to put into their wallets and purses to carry around with them. Because they're educators, and many of you will be as well, to educate people on the value of vaccination. There are a number of ways to do that. If I were asked to do it, I would show people this picture. Look, this red line represents what your body can do. But if you give your body the ability to make these dormant or memory cells, look at what it can do the second time it sees it. The power is in the secondary immune response. This is the way I would try to convince people that they ought to vaccinate their children. All right, let's talk about antibody structure for a few minutes. We've been looking at them as being produced by the B cells, but we haven't talked about their structure. An antibody is a protein. And there are about 5 different varieties I'm going to talk about in the next picture and talk to you a little bit about where they're found. But antibodies themselves can be monomers like this, single quaternary proteins. They can be dimers. They can be pentamers. Some of them, 5 of these all stuck together. And I'm not going to teach you all of that. It doesn't matter to me that you can describe the structure of each of the different antibodies. I would like for you to be able to be aware of their general structure, which is what this picture is. And I'd like you to be aware of the major varieties of these that are already present in the plasma. All right, so first, this is the general structure of an antibody. It has 4 chains. Hence, we say this protein has quaternary structure. That's a protein, that's a protein, that's a protein, and that's a protein. These protein chains, these polypeptide chains, are stuck together by cysteine residues, specifically by the sulfur residues and these cysteine residues. These are called the all-important and famous, organic chemistry people, molecular biology, the all-important and famous disulfide bonds. Disulfide bonds hold the antibody molecules together. They hold the chains, the 4 chains of the antibody molecule together. This is prototype. Cysteine residues and disulfide bonds. Now, of these 4 separate polypeptide chains, there are 2 little ones and 2 big ones. The 2 little ones are on the edges, and the 2 big ones are on the interior of the protein. You can see that. Of each of these polypeptide chains, there are regions of the chain, the solid regions here, that are constant. That is, they have the same amino acid sequence. So this one is the same as this one, and this one is the same as this one. In fact, if you compare these constant regions to the other types of antibodies, you would find that they are the same as well. So here's the thing. In your body, antibodies that are produced by B cells in your body all of them have similar amino acid sequences in the constant regions. You follow that? Same here, same here. It's a dimer, pentamer, whatever the antibody is, constant regions are the same. Now that really flies in the face of what I said a few minutes ago, and I said when I first introduced immunology to you. It flies in the face of this fact. The recognition capacity is already there for essentially any antigenic sequence that your body would come in contact with. Having the same amino acid sequence does not help me explain that. It does not make sense. However, the tips, the ends of these constant regions and the antibody molecules does help. These are called the variable regions, and these are the places where the antigen binding sites are located. So the tips of the antibody molecules are variable regions, and these are the places where the antigens are bound. This, y'all, the amino acid sequences here in the variable regions is where all of the explanatory power of how antibodies recognize antigens is here. So if you want to track backwards in the system and try to figure out how the immune system got so good at what it does, you have to track back to the genes, the messenger RNAs, the transcripts, transcription factors. You have to track backwards in the system to find what the sequences of base pairs were that caused the sequences of amino acids to create these variable regions. So you can see how gene regulation plays such an important role here. We're going to talk about how these genes produce these proteins. That's where the power of immunity is. And there are two big schools of thought, and they fight with each other in the literature as to how this happens. I'm not an immunologist, but I know enough to be dangerous. And so when I read what these people say about each other, it only demonstrates one thing to me. They don't know. If there are two schools of thought and they don't agree with each other in explaining how you get a variable region on an antibody, then what can I conclude as a basic scientist? We don't know yet. There are different ideas about how the genes are regulated to produce these variable regions. So look for it later in your life. Listen to Dr. Criswell's description of this and how the variable regions are produced. I wish Trenda was here so she could back me up on this. It's very interesting, and it's not really a point for physiology. I just want to point out to you the variable regions and try to get you interested. Antigen binding sites. Now, there are several varieties in circulation. These are the gamma globulins. 3 groups of dissolved plasma proteins: albumins, globulins, fibrinogen. Remember that? The albumins, carrier proteins that also are responsible for osmotic pressure. Globulins, we call them alpha, beta, and gamma. The gammas are the only ones we're talking about here. The gamma globulins are the circulating antibodies. And so here they are in variety. There are 1, 2, 3, 4, 5 varieties. Now you say, okay, now wait a minute. Wait. So you just said, Wade, that essentially any antigen that I can be exposed to, I already have variable regions to recognize it. What do you mean there are only 5 types? Can y'all do that? Can you work through the logic of that? In fact, let me make it worse before you try to explain that. 80% of the circulating antibody is IgG variety. 80% of it is IgG. So what I'm telling you is the vast majority of the circulating antibody is of one type. How is it possible to be so diverse, but yet be of one variety? Now it's your turn. How is it possible that 80% of the circulating antibody is of one variety, but yet it has so much specificity that it could recognize essentially any antigen that I was exposed to? How's that possible?
I don't know. I'll say the variable region of the IgG is undergoing a lot of recombination process.
Yes.
And like different recombination process also like give them the ability to recognize everything. And also I think they're lighter, that's why they can travel into the system. And the IgM, they're usually dimers. And
That is correct. Yeah. So you've had some of this. Did you take immunology already?
yes.
Yeah.
So why did you start with I don't know?
Because I'm not sure.
Yes.
Well, I'm not pretty sure.
You sure about that?
Yes. So he's absolutely correct, and this is why I introduced this by showing you the variable regions. The answer is here.
The regions vary.
The regions vary. Yeah, exactly. So just because I say that it's all IgG, I do not mean that it's not powerful enough to recognize lots and lots of different antigens. Okay, so we split them up. These are split up by weight. And so the very famous way to split up proteins in molecular biology is with the use of a technique called Western blots. And you can separate these guys out into 5 different bands. That's why they're here. And as Jancy's pointed out, some of them are dimers and some of them are pentamers. And so they have different structures. They have the same base structure, but they have different weights. Okay, So the IgG variety is good for bacteria and viruses, and look at this. It activates the complement. Now, I haven't spoken about complement. Most textbooks include the complement system in the non-specific defense mechanisms. So I gave you species resistance, mechanical, enzymes, interferons, fever, inflammation, phagocytosis, non-specific things. I did not put complement there. Most textbooks do. The reason I don't is because I like for my students to think about the complement system as being a next step beyond specific defense. That is, the complement really needs the specific defense system to work. And specifically, it needs the IgG populations to activate these complement proteins. Now, there's a bunch of them that we know of now, about 30 of them. The first 9 create this incredibly elegant complex in the membranes of cells called the membrane attack complex, a very famous thing in immunology called the MAC, the membrane attack complex. And I'm not gonna make you. I have in years past made the students learn about 1A associating with 1B and how that plugs into 1C, and then it creates a hole in the membrane. And so I don't see with the point of that to drag you guys through that. I do, however, want you to know about the importance of the complement proteins. And so I have a separate slide to talk about some of the really cool stuff that complement proteins do. They're activated by the IgG class of antibodies. The IgAs are found in breast milk and tears and gastric juice and bile. The IgMs, we've already seen. These guys are the antibodies that cause agglutination reactions and transfusion reactions. So anti-A and anti-B and anti-D, these are IgM class. IgDs, I never talk about because I don't know much about them. They activate other B cells, and so I've never spent much time in this group. But I will finish the presentation here today with the IgEs talking about allergies. So type 1 immediate onset allergies are facilitated by these antibodies, the IgEs, and they're binding to mast cells. So that's where we'll finish that. Now, here's the complement story. IgGs activate complement. So what are some of the things that these complement proteins can do? All right, so here's my list. 1, 2, 3, 4, 5 things that complement proteins can do. So terminology. First, they can perform what's called opsonization, enhancing the phagocytosis of antigens. I like to think, so I put little doodahs on these, and maybe they're not all appropriate, but it helps me remember them. Opsonization makes what the phagocyte sees taste good. I like to think about opsonization as smearing the butter on the bread. I'm going to eat this bread anyway, but you smear some good old-fashioned butter and jelly on that thing, I'm going to really get after it. So I like to think about the butter and the jelly as the complement protein opsonization, enhancing the phagocytosis of the antigens, making them taste good. All right, here's the second one. Complement proteins can cause chemotaxis, attracting macrophages. Okay? So if opsonization is spreading the butter on it, then chemotaxis is dropping the butter in the black iron pot before you start to cook. So you come into the house, and if somebody's cooking something, if they've dropped the butter in the pot, you don't know what it's going to be that you're going to eat, but you know you're going to like it because it smells good. You have been attracted to it. You follow me? So I use butter for both of these, butter on the bread and butter in the pot. Opsonization, enhance the phagocytosis, make it taste good. Chemotaxis, attract them, get them into the area. Complement proteins can do both of these. All right, this is the one that I used to teach the details of, and so I'll just mention it here. I would like for you to know the term at least. It's an important immunology term. Lysis. Lysis is a primary way that complement proteins get rid of things. And they do so by forming holes in the membranes of cells that are infected. Now, does that sound familiar in terms of strategy? Remember the cytotoxic T cells or the natural killer cells? They release perforin that cuts holes in the membrane. In this case, the complement proteins actually form the holes. They embed into the cell membrane and form, here's the term I'd like you to remember, they form what's called the membrane attack complex, the MAC. And it is really nothing more than a pore that water can go through. And so as it forms that watery hole there, that hole the water can pass through, water will pass into the cell and it blows up. This is the way they destroy the cell. We know that complement proteins can alter the molecular structure of viruses. You're going to have to ask Dr. Criswell about this one. And we know that complement proteins can cause some clumping of the antigen-bearing agents. It can cause some agglutination. They can do some of that on their own. So here are the 5 big things that complement proteins are capable of. What group of antibodies activate these? The IgGs. So when I think about the sequence of the immune system, when I think about, okay, let's go through the process. In my mind, I go, it's in me and I don't want it. So I'm going to use phagocytes to engulf it. So I want to vacuum it up. It's in me and I don't want it in there, so let's clean it up. So I vacuum it up. And when I vacuum it up, I want to take little pieces of whatever this is that I don't want in me, and I want to show this to the rest of the system so that it knows what to be looking for. After I've done that, after I've shown the rest of the immune system what I'm looking for, I want the specific guys that target these specific pieces to go after it. After these antibodies and stuff have gone after what it is, so it's looking all over the place for it now to get after it, I want as a last step here to produce a system to activate a group of proteins that will help me ultimately rid the body of it, to destroy it. This, to me, is the last step, the activation. The IgGs activate the complement that go after it in the last step. So whenever I think about the immune system, I always think about the CD4 story. Macrophage to CD4 to B cell to IgG to complement. Y'all see the sequence? That's the way I normally think about it. All right. Now the last thing I'll bring up, there are most texts have 2 to 4 different varieties of allergies they talk about. And so we'll just talk about a couple. I like to bring this up because I showed you the IgE population. So this is the mechanism of an immediate reaction allergy. And as you can see here, it involves the release of the IgE family of antibodies. So up here you can see, here comes an allergen. You can see that the variable region of the antibody has recognized this thing. Tell me, is this a B cell activated by cytokines? Or is this a B cell acting alone? It's acting alone. You see that? So it has its own variable region here to recognize the allergen. And when it does, it releases its IgE family. You think, okay, well, it's a plasma cell now. That's what they do. B cells have now acted by themselves. They're going to release these antibodies, and these antibodies are going to go out, and they're going to recognize these allergens. They're going to bind to them. They're going to help to clump them. They're going to bring on the complement system to get after them, whatever, to get rid of the stuff. Okay, true. But as an interesting piece of molecular anatomy here, we have within most of the connective tissues of our body high populations of mast cells. Mast cells, for reasons that you will never be able to explain to me, have binding regions in their membranes that recognize the constant regions of the IgE class of antibodies. Now, if these mast cells did not have binding regions for the IgE antibodies, there would be no such thing as allergies. They would not exist, at least not the type 1s, the ones that we most often think of. snotty nose caused by the pollen. This would not exist if the mast cells in our tissues all over our respiratory system did not bind the IgE class of antibodies. So here's what happens. After the IgEs bind here, they sit there in the mast cell. And if there is a subsequent contact, a subsequent exposure to the allergen binding to the antibody on the mast cell facilitates the release of granules in the mast cell that contain a very harmful molecule called histamine. Now there are also others like the leukotrienes that are released here too, but the one that we know is doing a lot of harm is this one, is histamine. The histamine released here can do a number of things. One of the most dangerous is the histamine can cause the contraction of the smooth muscle of the airway. And in doing so, it can shut down your breathing. You can die. So that description of an immediate reaction allergy would be the description of an individual who is hypersensitive, who has a hypersensitive immediate reaction allergy. And in that case, these people are going to carry around drugs that are going to help them open the airway after the histamine is closed it. And you guys know what that drug would be. Epinephrine. Because it's going to bind to beta-2 adrenergic receptors on the smooth muscle and cause them to relax. And this one's causing them to constrict. Now there's about a half dozen different histamine receptors. We're going to talk about another variety in the final section of this class. H2 receptors that are found in our digestive system that we need to make acid with. So antihistamines can also be acid reducers, as it turns out. But for today, the histamine can be a problem. Now, let's see if we can answer a question. We know basic biology now. We know basic immediate reaction allergy things. We get fundamentals here. Now, let's see if we can answer some questions. Here's my question for you. Why is it that some people can be stung by a bee and it hurts and it swells up, but then they get better and they never have any problems with their breathing? Whereas other people could get stung by a bee and within moments, if they don't have epinephrine in their system, they'll die. Does this happen in some people and not happen in others? Is there any chance that your immune system would not recognize the bee venom? Is there any chance that your immune system would not recognize peanut butter as one of the most common allergies today? Or any other number of things? Is that what's happening? Yeah? Well, I thought your teacher just said a moment ago that the antibody populations in your system produce variable regions that can recognize essentially any antigen that we would expose our system to. Why is it that it doesn't bother me, it bothers you?
Is it because not everyone's variable regions, when they recognize it, will activate the B cell to become a plasma cell?
Okay, keep coming, Hannah. I think you're really close here. So, say that again.
So, when some people, when they're exposed to the allergen, the B cell is going to recognize it with its variable region because it can recognize almost anything.
Okay, so let's stop there. So, I asked the question, does your immune system recognize it? And I would say, yeah. It knows it's there. You normally get a little inflammation there or something. Your immune system knows it's there. All right, so keep coming.
But the reason that not everyone will, like, their throat closes up when they get stung by a bee is because not everyone's body will let the B cells become plasma cells.
They don't elicit such a strong response to it
Okay, could you tell me why that would be? Why
because
would,
it doesn't determine it to be a life or death situation.
well, so the reason for this is because of the amount of this stuff. It's how much of this stuff is present on the mast cells. That's really the reason for it.
So they have less of the IgE receptors on their mast cells for peanut butter than someone who is severely allergic.
That's a good way to say it. I like that. So that's one reason. Okay, so less binding domains, less mast cell binding domains. That's one reason. Another reason, it's not usual for people to have to make enough of this to be a problem. So look back at this. See, I've got 80, 93, and 6 is 99, and so I've got two more versions of this antibody here to make up what percent of the dissolved plasma protein? Yeah, very, very small titers. So what would you say then about the person who is in danger here with the titers of the IgEs specific for beast venom or peanut butter?
They have abnormally high amounts of receptors.
Why would that be?
Maybe they've been exposed before.
Okay, it could be a previous exposure, and that does complicate the matter sometimes.
Their body doesn't have enough of the other ones, and so it makes
Okay.
IgE in place of it.
Okay, I don't know if that's true or not, but I do know that it has to do with their genetic makeup.
That's what, is anything hereditary?
Yeah, well, so there is some hereditary component to allergies, that's true. But what I'm thinking here, the more basic explanation for this is just simply the genetic makeup of the individual producing the antibodies. That is, I have a family of IgEs in my system, and there are a number of variable regions on them. You have IgEs in your system. There are a number of variable regions on them. They recognize a plethora of different antigenic sequences, peanut butter and bee stings being in the group. Now, something in the genetic makeup is putting together the variable regions. What can you say about the genetic makeup of the individual who is going to be life-threatened in the makeup of the variable region?
They make a lot of variable regions. that
Yes!
exist.
Y'all see that now? That's the answer. The answer ultimately is their genetic, whatever the recombination is, Chance, that you were talking about, however you put those variable regions together, is done in a way so that the specific variable regions specific for that antigen are overproduced in your system compared with everybody else. It doesn't necessarily mean that you have a higher concentration of IgE. It means you have a higher concentration of IgE with variable regions that recognize that allergen. The only way that I know of today to do this is to expose people to it. They stick people with little, they pinprick people with little pieces of these antigens to see. Because if you have a response to it, you're going to get a little inflammation right there where the allergen was. Very interesting to me. And it's one of the reasons I like to bring this up, because knowing about the variable regions of the antibodies can really help us explain these kinds of things as to what's going on with people.
So you said this was a type 1 allergen.
Yeah,
So there's more types of allergens than just this process?
yeah. So this is type 1 allergies are typically a B cell phenomenon. They're a histamine release issue. If you want to go after a type 1 allergy, you give people antihistamines. The sprays that you put in their nose, there are now, people can take allergy shots for these type 1 allergies now. And these shots are really designed like Rogam to hide the system, to hide these things, or to produce ways to destroy them. I don't know how all the allergy shots work, the different strategies on different ones.
So
But there are, let me finish that. So the other major group of allergies that people know of are things like skin problems, poison ivy, poison oak. You touch something in those oleoresins that are organic get through your skin and you get this rash or something on your skin. Well, you treat, these are B cell phenomena. The phenomenon in your skin when you get a rash is normally a T cell. It's a cell-to-cell kind of mediated action. Langerhans cells present in the epidermis, immune system cells that do this, and activate T cells in the area to release inflammatory cytokines. The way you go after that is not with Benadryl. You go after those with corticosteroids. Cream. What do the creams contain in them? Glucocorticoids. Cortisol. And what do you guys know cortisone does to the immune system cells?
It suppresses them.
Cortisone, it suppresses immune system cells. So that's why the cream works on the rash. It is suppressing the activity of the T cells there. So I normally think about, when I think about allergies, I think about them in those two broad categories. It's probably a little oversimplified. B cell phenomenon, histamine. T cell phenomenon, skin conditions. But there are others. I think that's type 1 and type 4. Type 2s are something with the respiratory, and type 3s are something with autoimmune stuff, like rheumatoid arthritis. I don't remember all the details of those, but the general categories, I bring this one up because it's antibody related. You had another question. I interrupted you.
No, so I know a little girl and she has a severe milk allergy, but she's allergic to a protein in milk. So is that a type 1 or is that a different?
No, it's type 1.
So if she has the oversensitive variable regions for a certain protein found in dairy.
Yep.
Okay.
That's what that is. And so if she drinks it, she can get respiratory problems quick.
Yeah, if someone's been drinking milk and touches her, she needs an EpiPen and a Benadryl.
Yeah. This little girl, guy used to teach communications here, Eliza Trice, that's her name. She's allergic to peanut butter. I didn't know. She's sitting on the counter, the activities building one day, and she's cute as a button. And I had a pack of peanut butter crackers, and she said, like this, what kind of, so I handed her a peanut butter cracker. And Stephanie Blair was standing there by her. And so Stephanie turned and looked, and just as the girl got her, and she slapped the cracker out of her hand. Like that. And what do you think the child did? Cried? Yeah. And I said, what is your problem? She said, well, you almost killed her. The girl is hypersensitive to peanut butter. And if she had eaten that, we'd have had to give her a shot of epi to save her life. I've got it right back here. Her mom gave it to me. I'm supposed to be watching her. Yeah? Type 1 allergies. Very dangerous. Okay. When we kick this up on Monday, I'm going to use histamine to get you into the respiratory system. So the histamine epinephrine story. We're going to start talking about breathing. So respiration is the last content for your next test. So I think we have 3 more class periods, a week left to do respiration next week.